go along. So today and tomorrow, we are going to start with Galaxy and cover it by tomorrow, right? So first, what I'll do is I'll start with some of the basics of genome sequencing. And then we start the analysis today and try and cover it by tomorrow, right? So because the Galaxy takes a lot of time to take the data and upload the data. So if you're doing it parallelly, you will have long pauses in between. So in that case, because I've already made the PowerPoint, we can cover it on the PowerPoint, and then you can match your data tomorrow, maybe, right? So let's get started, and we start first with some of the basics of NGS, and then we move on to trying and understand how to analyze NGS data with respect to whether your genome contains genes for antimicrobial resistance or not, right? So that is where we'll use Galaxy. So first, let me share my screen, and then of course we move on and we talk more about NGS. So here you are, and let me share my screen now. So let me share this here. And I hope you guys have been doing some R after we had the initial lectures, because again, let me repeat, it is important that you keep doing everything, right? So if you don't do programming, then you're going to miss on it. So how many of you have been doing a bit of R after we have met the last time? Anybody who has been practicing a bit of R?
Sir, I did, but graph was not able to plot, but that Doppler plot you told about that.
Graph I had told you through ggplot2, and in case you're not able to plot, you could have gone back to my videos and checked. I'm sure there must be some typo or something. Else you can send me your screenshot. I'll have a look, right?
Yes, sir.
So
So share the email ID. I will share that.
email ID is given on my website, but anyways, I'll share my email ID otherwise also,
Okay,
right?
thank you, sir.
So that should be fine. Okay, so let's get started, and we talk of, and Bhavya says she practiced, so it is very nice. And there was a long message I missed. Manisha says no, so if you don't practice, it will be a problem. But then for those who find programming a little difficult, the answer is Galaxy, right? So in Galaxy,
No, sir. Actually, I answered to the question that is written over here. Have you all got the day before yesterday's workshop video?
Oh, okay. I'm so sorry. Right.
Okay.
No problem. So let's get started, and we talk first of a bit of NGS technique as such, and why it is important to go through these steps before you can finally get a feel of your data. So here you are, and we will talk of yes. So here you are, and yeah. So first, let's talk of basic concepts in next generation sequencing. So what we're going to cover today is basically next generation sequencing and a bit of Galaxy. So we are talking of a journey of 20 years, starting from the Sanger sequencer, which was the first left-hand corner. Then we moved on to the second generation of sequencing techniques, which largely covered Illumina and Iron Torrent. And now we are into the third generation of sequencers, which is basically the long-range sequencers. We have MinION, and we also have this PacBio machine that can basically single molecule real-time sequencing. So we'll talk a bit of these and then come back to our data and data formats. And then we come to Galaxy and how this can be used to analyze your Galaxy, to analyze your data, right? So what is sequencing? I'm sure everybody knows what is sequencing. Sequencing essentially means to determine the order of nucleotides in a given nucleic acid. It could be RNA, it could be DNA, but generally what we're looking for is the order of nucleotides in a given nucleic acid, and you're looking from 5' to 3' in with respect to the orientation, right? So this is sequencing. I'm also sure that you know what is a genome. So one full complement of genetic material in an organism or a cell is what is known as a genome, and sequencing genomes is challenging. So the question is, why is it so challenging? We'll talk of it as we move along in the presentation. So here you are. Why do you want to sequence genomes? Well, the answer to this is simple. We want to understand the basic composition of the human and other genomes. So because DNA is the blueprint, so we want to understand what is the basic composition of the genome as such, because this is the blueprint, this is what governs everything else. So if you go back to the central dogma, you have DNA transcribes into RNA, and RNA transcribes into protein. DNA is your blueprint, RNA is your intermediate, and then of course you have the protein, which is your workhorse. So mostly for most organisms, this is the central dogma. Of course, in retroviruses, your RNA is the genome. So because the entire thing that you have is encoded within the DNA, it is important to know what is the order of nucleotides in the DNA, right? Then, of course, what is also important is you want to identify what are the variations that are there. And more importantly, we are looking for biomedical applications, and we were looking for variations that cause disease, right? So that is where our interest in genome is. And of course, for microbes, we're looking for whether a microbe contains antibiotic resistance genes or not. That would allow you to then, have some counter strategies against it. So this is where sequencing is. And of course, sequencing started way back in 1977. You sequenced 5X174 virus. And then beyond that, 2001 was a major year in sequencing because we were able to sequence the human genome. And since then, there has been major developments in sequencing. The last 20 years have been remarkable. And now, of course, you can sequence the human genome in less than half a day and at around $1,000, right? So this is where we are heading now. So our story begins here. We are talking of February 2001. This was a red letter day in terms of sequencing. We had the announcement of the decoding of the book of life, the announcement of the human genome, and human genome was sequenced by a company called Celera Genomics, which was headed by Craig Venter, and a publicly funded organization called Human Genome Organization, which was headed by Francis Collins at that point of time, right? So this is where we begin. And then, of course, since then, it has been a journey, very remarkable, most remarkable, so as to say. So as I mentioned, that genome sequencing is a bit complex. So now we talk of why it is so complex. So the idea is that even today, we do not have an enzyme that can directly start from one end and, sequence the entire DNA in one go. So when you have a bigger problem, what you do is you break it into smaller pieces. So here in the red line, I show you a genome that has these fragments, which has this order. So you have the initial part is A, D, C, B, and E. So what you do is, because you cannot have an enzyme that can directly sequence from 5' end to the 3' end, you break it into smaller pieces, right? And my mouse is not working. Yeah, so here you are, you get your pieces, and then you individually sequence these pieces. So you have the sequence of A now, you have the sequence of E, you have the sequence of D, you have the sequence of C, and you have the sequence of B. Now the challenge is to rearrange it back in the order you have in the original genome. So that is where the main challenge is. The main challenge in the assembly. So if you look at the steps of sequencing a genome, first you have to fragment into smaller pieces. And this will depend on the platform that you're using. So nowadays you have two types of sequencing platforms. You have short read sequencers like Illumina and iTorrent. Illumina is the market leader as of today. And then now you also have in the market what is known as long read sequencers like Nanopore or SMRT that can directly read very long pieces, but not so long enough to be able to sequence an entire genome in one go. Class, you're following, everyone?
Yes, sir.
Okay.
Yes.
So this is what you do. So basically, whether it is a genome or a problem in life, the way to go forward is to break the problem into smaller pieces, find individual solutions, put the solutions together to get the final solution. So this is what we do. We first break the genome into smaller pieces. How small a piece it would be would depend on what type of platform you're using. So if you're using Illumina, right now Illumina gives you around 150 bases of sequence. So you have to have a fragment around 150 bases. If you're using Nanopore, very easily it can give you up to 10 kb sequences, so you can have fragments of 10 kb. Likewise for PacBio. So then, of course, you sequence the fragments. So you have the sequence of A, B, C, D, and E. And then comes the large and the most challenging part, last and the most challenging part. This is to arrange the reads in the order they appear in the genome. So you have the full sequence of the genome, but in parts. You now need to arrange these fragments together to get the final genome. So this is how your final arrangement has to be. And then you stitch them together to get the final full sequence. So this is where the challenge is. And while we are now having long range sequences, there's still some part that remains to be solved. We still do not have an enzyme that can directly start from one end and sequence the entire DNA if it is too large. Then, of course, so because you have fragmented your DNA, so when you're assembling it, you generally do a reference-based assembly. You could also do a de novo assembly, but here I show you a reference-based assembly. So what you're doing is this is your reference sequence that you already have, let's say the human genome, and these are the reads coming from my genome. So my genome has been sequenced by Illumina, so I have these small reads. So what I do is I align these reads back to my reference genome. So this is your reference human genome. I'm aligning my reads to the reference human genome, and I'm looking at each position, each nucleotide. Let's say chromosome 5, 500. So I'm looking how many of these reads got aligned to this position here, right? And I'm looking at this position. So I say this coverage here is 8x because 8 reads at this position have the nucleotides at this position here, right? Likewise here, there is only 2 reads that align to chromosome 5 position 5000. There is no matching in this region, so there will be a gap and so on and so forth, right? So coverage is critical in variation and epigenetic analysis. The higher the coverage, the more the number of reads you have, the more confident you are of calling a base at each position, right? So that is important again. So the higher the coverage, the more statistical power and confidence in the analysis you have. And coverage typically is, for example, here, so you could have what is the coverage here at this point class? 1, 2, 3, 4, 5, and 6. So this would be 6x, right? And then of course here there is no coverage, so there is no read that is aligning at this point. So you will have a gap in assembly when you do your assembly. So you'll have all these reads joined together into one contig. You'll have all these reads joined together into a second contig, and you'll have a gap in between, right? So this is what you have. And if you look at how do you calculate coverage, so coverage at each position can be calculated simply by the formula C equals to small n is the number of reads aligned at this position, and then into the length of the each read. So this is a small read length, and divided by the total genome length, right? So this is fixed, capital L is fixed, small l is also fixed. All that you can change is the number of reads aligning to the region. So therefore, the more number of reads you have, the more higher the coverage, the more confidence in data you will have. So with Sanger sequencing, you had generally a coverage of 10x or at max 30x. With next generation sequencing like Illumina or for that matter others, you can have a coverage of very easily a coverage of 100x or 150x. That depends on how much money you have in the project and how much you can spend on the coverage. So the higher the coverage, the more the cost of sequencing you'll have. Right? Everybody here, class? You're following?
Yes, sir.
Yes, sir.
Okay, very good. So that's a confident test. So then we move on, and then we talk of the second important concept in next generation sequencing. This is called read length. So this is an image of Eiffel Tower, which I took at Châtelet in Paris when I was doing my postdoc. So here is the puzzle that I'm giving to you is that you have to break this image into parts and then reassemble it back into the original image. So you have these many parts here. Let's say so you have 4 parts, you have 9 parts, and you have 56 parts. So which one will be most easiest to assemble?
4 parts.
A, B, or C, right? 4 parts. So very rightly said, right? So likewise in genomes, because you have lesser number of parts here, this is easily assembled and there is less chance of going wrong here as compared to this image here, where you can very easily, misassemble this part for this part, and you can very easily misidentify this part to be here versus here, and you could swap places here. So this is what is the concept of read length. The smaller the read length, the more difficult the assembly, the more chances of error, right? So here you are. So this is what is your, let's say this is basically representing your library. This is a 4-part library. This is a 9-part library. This is a short read library with multiple parts, 56 parts. And then when you assemble back, the shorter the reads, the more the chance of creating an error while assembling, right? So this is again something important, and more so with the complex genomes as humans, because human genome, as you know, has 50% repeat content. So therefore, it becomes even more difficult to assemble it together unless you have a reference. And that is one of the reasons why it took almost 10 years to sequence the first reference genome. And after that, because you already had a reference, it became a little easier to assemble the genomes, because it's like going to a place with a Google map and without a Google map, right? So here you are, the concept of read length. Read length is the maximum sequence length that can be obtained in one sequencing reaction, and this is specific to each sequencing platform. So depending on which sequencing platform you are talking about. So for example, Illumina will have around 150 bases, Iron Torrent 100 bases, Paris sequencing, which is now obsolete, had around 400 bases. Sanger sequencing had around 900 bases, and so on and so forth. And then of course you have the long read sequencers like you have your nanopore and PacBio. These can give you 10 KB of sequences and so on and so forth. We'll talk of nanopore as we move along. because nanopore data is what we're going to use for galaxy analysis. So read length is a critical parameter for completeness and accuracy of the genome sequence, more so in repeated genome. So therefore, now the trend of sequencing is towards the long read sequencers. All right. So now, if you look at the steps in NGS data analysis, you have basically, if you look at first, of course, when you get your NGS data, you have to go for what is known as data quality control. And if you see that your data is slightly, not up to the mark, you can do some modifications like trimming or removing a part of the sequence. If it has adapter contamination, you can remove the adapters and so on and so forth. And then, of course, the next step is to go and map it onto a reference genome so that your reads can be mapped onto a reference so that you have the assembly in a proper way. And then, of course, beyond that, depending on what kind of analysis you're doing, you have several different types of analysis. The 5 major types of analysis that you can do with sequencing data include DNA-seq, where you're looking for variations in your DNA with respect to reference. So you assemble your data and compare to the reference. You could be looking for the transcriptomic data, and you could be looking for your RNA, your gene expression levels. So you do an RNA-seq, which is here. And you could do a differential gene analysis. You could be looking for protein DNA interactions, which is your chromatin immunoprecipitation, and then sequencing NGS based. So this is called ChIP-seq. You could be looking for your bisulfite data. This is called bisulfite sequence because you treat your DNA with bisulfite because you're wanting to look at the methylated cytosines. So this is kind of epigenetic sequencing that you do. And then you could be looking for chromatin state. So you do what is known as ATAC-seq, right? So these are the 5 major analysis that you can do. And of course, there are variations to this, and there are a lot more type of seqs that you can do. For example, fire-seq and so on and so forth. So we'll not go into the details of that. We are largely confined here to DNA-seq because we're going to read the bacterial genome and then assemble it into the final sequence and then look for genes that whether it contains genes for antimicrobial resistance or not. So that is going to be our basic data. And because the data is huge and of course it is voluminous, manual analysis of this data is not possible, and you need to know a bit of programming know-how. And if you don't know a programming know-how, then platforms like Galaxy, which are like workflows and pipelines, they come to your rescue, right? So that can also be done using Galaxy. So we'll discuss Galaxy as we move on, but first let us have some basic idea of the data analysis part here. And the data formats become important here because everything will be, even at the backend in Galaxy, there is a program that is running. So a program recognizes a specific format. So the data has to be in a specific format for it to be processed further. Otherwise, you'll have problems in your analysis. So therefore, we'll talk a bit about data formats before moving on to Galaxy. So here you have NGS data analysis and NGS data formats. So first, let's talk of the data format. So this is, can you see the slide title? Anyone?
No, sir, it's okay.
Can you see the slide title or not? Because on my case,
yes, sir, it's visible.
Okay, then it is fine. Because in my case, it is coming on top. The bench for WebEx is coming on the top and hiding the title. So this is what is your fast text, right? So this is what is your fast text here. It says paused. Can you hear me? Resume.
Yes, sir.
Okay.
Yes, sir.
So here you are. Okay. So here you are. So this is the first step in data analysis. And most commonly, if you're using the Illumina reads, you first go for what is known as a fast QC program that can give you the quality of bases at individual positions in your reads, right? And there are many other graphs that you get, but usually this is the most common plot that you get at the top and the first plot, which tells you what is the quality of individual positions. And initially, you'll have individual positions 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then you'll have binning. So this is 5 positions given together, and then it is the average quality of the nucleotide called at this position. And these are typically your box plots. If you have seen my videos on R, you would know that the box plot is basically a 5-point summary of your data. It tells you the distribution of data in terms of the first quartile, the second quartile, the third quartile, and then the interquartile range, and then the final maximum minimum and the outliers. So this is basically one part. So fast QC aims to provide a simple way to do some quality control checks on raw sequence data. It provides a modular set of analysis which you can use to give a quick impression of whether your data has any problems, of which you should be aware before doing any further analysis. So what is this? This is basically your, on this scale here on the y-axis is your Fred score, right? So Fred score is one way of assessing what is the base call quality, and anything below 20 is not generally acceptable. If you see, there is a green line here. From 28 to 40 is what is acceptable. Other than that, if you have some values that are below 20, it is a problem. So generally, you need to redo things. And what has been commonly observed is that at the start and the end of the sequence, there is generally a fall in quality of the base call. So those parts could be because of the adapter contamination, could be because of the artifact of the machine. So therefore, those have to be trimmed. So this is what you do. Yellow box is the inner quartile range for 25th to 75th percentile. The upper and lower whiskers represent 10th and 90th percentile score. So this is the whisker box plot here. Then we move on and we talk more about other things. So let's get on. So then, of course, as I said, once you have done your fast QC, you have trimmed your data, you have to basically align your data to a reference now, right? Or you could do a de novo or sequencing of the assembly. Most commonly with short reads, you do a reference-based assembly. So you align your data to a reference. The common read aligners include Bowtie, which is the second version now. It was earlier Bowtie, and now it is Bowtie 2. You can also use Burrow-Wheeler's aligner. You have his HiSat2, which is also part of Galaxy, Mummer, which is an HMM-based aligner, Star, and then you have Top Hat 2. Top Hat 2 is for RNA-seq, and so is a Star, most commonly used for RNA-seq analysis. And then for nanopore data, you can use Fly, right? So this is for long read aligners. You have Fly, and we'll use Fly in our analysis as we go along. So one of the most, read alignment is one of the most resource intensive steps during NGS data analysis. So one of the most resource intensive during NGS data analysis is the alignment of the sequence reads to the reference genome. Therefore, it is a common question about which tool you would be best for you. All of these will give you roughly the same kind of results. It is a matter of choice, and it is also a matter of what suits your data more. But most commonly, what is used is Bowtie for Illumina and Fly for long read sequencers. We'll use Fly for our analysis. So what is happening when you're doing your sequencing, when you're doing your alignment? So here is your reads that you have. So you have these different reads, and when you align it on the reference genome, these would align this way. And if you have a repeat in your sequence, this will go and align to multiple places in your reference genome, showing you that this is actually a repeat sequence, right? And that is where the complications come. Then you could have also what is known as, so this is a single end sequencing where all fragments have been sequenced from just one end. You could also have what is known as a double end sequencing. So you basically create your library and you sequence them from both ends. And depending on what is your platform, let's say Illumina, so you'll get 150 base pairs from the front end and 150 bases from the other end, right? This is more like your back end sequencing. I'm sure you would have read how the human genome was sequenced. So one of the strategies was to use the back-end sequencing. So while backs were having more than 150 to 200 KB of insert, you could get 900 base pair from one end and another 900 base pair from the other end using Sanger sequencing. So these were used to align your sequences together. So here you are. This is your paired end sequencing. So let's say, for example, you know that read 1 and read 2, your fragment size is 400 bases. And when you align them together, what you find is that your read 1 and read 2 are 700 bases apart from each other, which means that there has been a deletion in your sequence with respect to reference, right? So this is what is the advantage that you have with your paired end sequencing. When you have paired end sequencing, it allows you to identify some deletions that may have happened in your sequence vis-a-vis the reference. So this is what allows you to basically identify your indels, or also it may allow you to identify your repeat polymorphism. So what could have happened here is that in between here in the reference genome, there is no allo repeat insertion, but in your genome, there is an allo repeat insertion. So what is an allo repeat? Allo repeat is a small interspersed nuclear element, roughly 310 base pairs of DNA. These are the most common repeats in the human genome, 1.5 million copies on an average per genome, right? And these are polymorphic, so they may be present in one person, may not be present in another person at that same position. And you could also generate how related two individuals are based on the repeat insertion polymorphism. So this is the basic idea here. If you have a paired end library, it helps you identify some of the genomic structural variation that may occur between the reference and the genome that you're aligning to. You follow class, everyone?
Yes, sir. Sir, can you repeat it again about LU insertion?
Yeah, so what happens is, there are transposons in the genome, right? And we are talking about a specific type of transposon that is called as retrotransposon. So retrotransposons move by an RNA intermediate and keep getting inserted into genomic positions. So ALU repeats are one type of retrotransposons. These are called as small interspersed nuclear elements. And these are the most abundant repeats in the human genome, roughly around 310 bases in size. And they're polymorphic, as in I may have it at a certain position, you may not have it because you're not related. But one of my parents would have definitely had that, unless it is an individual personal insertion in my case. Otherwise, what I inherit as an ALU insertion would be from my parents, one from the maternal chromosome or from the paternal chromosome. So therefore, what is happening here is, in this case, you have this fragment of 400 bases. So you know that you have a 400 base pair library, and you have sequenced the first 150 bases from one end, and you've sequenced the other 150 bases, the blue, light blue here, read 2 from the other end. So these are 400 bases apart in your library. But when you align it to the reference genome, what you see is there are 700 bases apart. So there is something extra in the reference genome that you do not have. So this could be an insertion or a deletion, or it could be a repeat insertion, right? So this is what is basically your idea of paired and repeats that can allow you to identify some structural polymorphisms or structural variations in the genome, right? So I hope we can now move forward. So here you are. So here is what we're doing. So how assembly complications can come with repeatants. So these are your individual fragments. The green ones are your unique ends. So you know that the fragment A has a unique overlap with fragment C, so there is no problem. But when you come to the end of C, this has overlap with B, also with F, also with E. So now there is a problem. So while you know that C follows A, after C, which of these could be the possible one that is next to it is not easy to discover because they have the same common ends. So this is where the repeat complications come in case of assembly. So therefore, you need a reference assembly to solve this out. And this is also where the long read sequencers come in handy, because when you have a long read sequencers, you would probably, span this region and come up to a unique region, and that allows you to identify the next pair very easily. So this is where the sequencing technology is moving forward now. We are moving from small read sequencers to long read sequencers. And in the next few years, the long read sequencers will be the only one that would survive. There is some problem with long read sequencers in terms of efficiency and in terms of accuracy. So therefore, what people now do is they have both the assembly is done both with some short read sequencers and also with long read sequencers. So you get your genome sequenced twice over with short read sequencer, also with long read sequencer. The long read sequencer allows you a compact and correct assembly, and short read sequencers help you overcome the errors that may be there at individual positions in case of the assembly, right? So that is the hybrid model that we're following now. Class, you follow everyone?
Yes, sir.
Yes, sir. So then we move on. So this is how you do. So this is your short pair and long insert mate pairs. So we have already discussed this. We'll not go into details of this now. Now we come to data format. Since the data is read by programs, pre-written scripts in any language, mostly Python, Perl, or R, the data representation must follow fixed rules of format. So why do you need a format? Because at the backend, there is a program that is processing it, which is trained to read data in a certain way. So even if one column is missing or one column is misplaced, you will have problems in your final data. So therefore, you have a format for sequence data. So sequence data, I'm sure you would know, is now represented in FASTQ format. FASTQ because the quality for each base is also included in the basic sequence data itself. And this is a 4-line format. I'll show you the details of this. Then you have alignment files also have a specific format, which is known as the alignment mapping format. And then the binary format of that, which is called the BAM format. Then you have the annotation files. They also have their specific format of data. So this is now the standard is JFF3 or GTF format. And then you have the interval format, so which is called as a BET format or browser extensible data format. We will not discuss this in details now, but I'm sure you can go back and refer to if you want to have a clear understanding of things. So here is the file that we Yes, Gaurav, you raise a hand.
Sir, I have a question, sir. Usually Galaxy also gives fast Sanger format.
Yes. So
Sir,
fast.
what is actually that?
So it's basically similar to FASTQ, not too much of a difference, right? And you can basically you have there's not too much of a difference. And if there is some, I'll cross check and get back to you tomorrow. They're still
Okay,
doing
sir.
Galaxy tomorrow, right? So I'm not
Okay,
sure
sir.
of what exactly is the difference, but I've seen FASTQ Sanger, it is mentioned,
Yes, sir.
right? Yeah. So I
Okay,
will
sir.
get back tomorrow. Yeah.
Okay, sir.
So here
Thank you,
you
sir.
are. Yeah. So here you are. So the idea is this is your FASTQ format. You have the first line, that is the identifier line. This is a unique identifier that you have, begins with the @ sign, and then you have a unique identifier. And that is basically, this identifier will not be repeated elsewhere in your sequence file. You may have tens of thousands of sequences, but this will be unique to this first set of sequence here. Then the second line is actually your sequence line. The third line has a plus sign, and sometimes it may not have anything else. Sometimes it may have a repeat of the first line itself, the identifier itself. And the last line is what is known as a quality scores line. These are encrypted quality scores. Basically, these are Fred quality scores, and these are basically, if you look at this bracket represents the Fred quality score of 29. The scale of the scores is around 0 to 40, right? And basically, Fred represents the probability of error that may have happened in calling, right? So a Fred score of 1. So we'll come to it and we'll talk more about it. So here you are. Each read is represented by 4 lines. @ followed by read ID is the first line. This is unique to every sequence that you have in the file. Then the second line is the sequence itself. The third line begins with the plus sign, optionally followed by a read ID. And then you have the fourth line, that is the Fred quality scores for each base call in the sequence. And this is exactly the same length as your initial sequence here. So if there are 60 characters or 100 characters here, there'll be 100 characters here as well. These are encrypted but they can be always converted back to actual values, right? So here you are. If you look at this, ah, so Fred is a program that assigns quality score to each nucleotide call in a sequence. Fred scores are logarithmically related to probability of error. So Q equals to 10 minus 10 log P. So Fred score of 0 means error probability of 1. So if you have a 0 Fred score, which means that it is absolutely sure, the program is absolutely sure that the base call is incorrect. A Fred score of 10 means error probability of 0.1, of 20 means 0.01, of 30 is 0.001, and of 40 is 0.001, right? So this is what you have. So if you see, the exclamation marks indicates the Fred score of 0, and capital I indicates the Fred score of 40, which is the error probability of 0.0001, which is likely to be correct, right? Which means that the error probability is so less that it is absolutely sure that the nucleotide called at that position is correct. So here you are, and then you have the alignment formats. This is basically your SAM file, sequence alignment map file, and you have several features here. This is a 11 column file. We'll not go into the details of this for now, but I'm sure you would be able to refer back and read it in more details. First, of course, is the query name, then you have a flag, then you have the R name, position, then the map quality, then you have the cigar string. Basically, this is to tell you the alignment and whether there are gaps in the alignment with respect to reference or not. And then you have the other details that are there. You can have a look at what these mean in your time. The idea is that you need to know all of this before you get into the analysis. Most commonly what happens, students get into the analysis part before understanding all of this, and that is where the major problem comes. When you finally get to the results, you're not able to analyze the results, right? So this is something that you need to know. Then there is one more thing that you need to know, that is known as the N50 value. N50 value is a standard value to tell you what is the quality of your assembly, right? So N50 statistics defines assembly quality in terms of contiguity, right? Given a set of contigs, N50 is defined as the sequence length of the shortest contig at 50% of the total genome length. So for example, here you have these contigs 100, 70. So this is the contig length. This is 100, this is 70, 60, 50, 50, 40, and 30. So the total length is 400. So we're looking at which contig, the smallest contig where you can get 50% of the total length of your genome. So that would be your 200. So if you say 100 plus 70 plus 60, so 70 and 60 is 130, 230. So 60 becomes your N50 value, right? Everybody, you follow class?
Yes.
Yes, sir.
Okay, so this is your N50 value. So N50 value, the larger the N50 value, the more you're confident that your assembly is good. The smaller the N50 value, which means that you had very small fragments which could be joined together to finally give your assembly, and therefore sometimes if it is too low a value, you may have to do your sequencing again, right? So N50 statistics define assembly quality in terms of contiguity. Given a set of contigs, the N50 is defined as the sequence length of the shortest contig at 50% of the total genome length. Arrange all contig lengths in descending order. So you arrange all the contig lengths in descending order, and then you look for the contig which gives you 50% of the total genome length, right? So add the contig lengths starting from the largest till the sum is equal to or more than the length of the genome. Equal to or more than half the length of the genome. I'm sorry, there's something missing here. The length of the contig where you stop counting is your N50. So this is where your N50 is, the length of the contig. So this is 60 is your N50 here. Remember, it is the length of the contig that we're talking about as an N50 value. So coming back now to our Galaxy, and we are going to start in a moment now, and of course we'll continue tomorrow. So you have two types of sequencers. You have short read sequencers as of today. So contemporary sequencing technologies, you have short read sequencers. So you have virtual terminator sequencing, Illumina, you have Ion Torrent, Roche. Roughly you get 100 bases from here and 150 bases from here. And then you have Nanopore, which is coming from Oxford Technologies, and this is on an average, it can give you 10 kb. It can give you even larger sequence reads like 30 kb, 50 kb. And likewise, you have smart SMRT, single molecule real-time sequencing from PacBio. The sequencing methods are different. Nanopore is helicase based. SMRT is polymerase based. So in SMRT, you are synthesizing a complementary strand and looking at what nucleotide gets added. In Nanopore, there is no such thing. We are directly pushing a DNA through a hole, and as the DNA passes through, depending on what is their A, T, G, or C, you will have different fluctuations in current, and that allows you to identify what nucleotide has passed through. Also, what is good about these two technologies, the long-read sequencers, is that you don't need your DNA to be specially treated to identify methylated cytosines. So epigenetic signature can also be generated directly without going through the complex process of doing bisulfite sequencing. So we'll come to nanopore now. So this is the idea behind nanopore is very simple. You have a nanopore through which there is this nanopore is set into an electric field. So there is a basal current that flows through the electric field through the nanopore, and you have this read once because there is nothing that is interfering with the flow of current. Then as you move in certain different sized molecules and differently charged molecules, there is fluctuation in current that happens. So here you are, depending on what shape and size and charge the molecule contains, you will have differential fluctuation in current. So that is a signature for what molecule is basically moving through the nanopore, right? So, and fortunately for us, A, T, G, and C produce different fluctuations in current. So all you need to do is to force a DNA through this nanopore here. And as the DNA moves through the nanopore, so what you have is a ratchet protein that forces the DNA through the nanopore. And as the DNA moves through the nanopore, based on what is passing through the nanopore, you have fluctuation in current. And this can be digitally converted into the actual nucleotide sequence at the back end. So this is marketed by Oxford Nanopore. You can sequence a human genome real quick here. And the size of sequencer is pen drive. So I've already shown you it can be held in a hand. So this is basically the advantage of nanopore. It can be used as a point of care machine. So instead of person coming to the laboratory for getting the sequence, the sequencer can be taken to the person. And for a country like India, where at remote places you do not have much of hospital facility, this could be the game changer, right? And then cost of sequencing for the human genome is now less than $1,000, and you can be on the lookout. We are very close to now announcing a genome sequencer that can sequence a human genome at $100, right? And this is where the actual next generation sequencing began. The challenge was to bring down the cost of sequencing from the original $3.2 billion for the first human genome, the reference human genome, to $1,000. And that is where the second generation and then finally the third generation sequencers have come up. So here you are. Let's move on and we talk more about this. So if you want to have a more elaborate lecture on next generation sequencing technologies, you can have a look here, and This is on YouTube again, my channel, Dr. Vipin's Biotech and Bioinformatics Classroom. You can have a look. I give a uh, detailed description of how the sequencing is done, what is the sequencing chemistry, and what are the applications as well, right? So, and I'm also giving a talk with Pine Biotech on Saturday on next generation sequencing. It is entitled From Evolution to Revolution, 40 Years of DNA Sequencing Techniques. All right, so now we come to Galaxy, right? So now the idea is, why do we need Galaxy, right? So first, sequence data that comes from sequencers is just raw data. It will not hold any, I'm sorry, it may not hold any meaning unless it is processed. So therefore, you need to have formatting and then quality check and then alignment and everything to finally find out meaningful inference from the data. So therefore, you need tools like Galaxy or online command line tools like the ones we can use with Linux systems. So to derive meaningful inference, information from raw sequence data must be processed, quality control, assembly, and annotation. A whole suite of tools are available, both command line and as graphic user interface, to assist NGS data analysis. Right? Galaxy is an open source, web-based platform for data-intensive biomedical research. So Galaxy basically is an open source. It is free for use. It is developed by people who are interested in serving the community, and it is a web-based platform for data-intensive biomedical research. We'll have a look at that. Galaxy follows the FAIR principles, which is now the standard principles for data analysis. So it is findable, accessible, interoperable, and reusable. So Galaxy is an open source platform for fair data analysis that enables users to use tools for various domains that can be plugged into workflows through its graphical web interface. Then manage data by sharing and publishing results, workflows, and visualizations. Then ensure reproducibility by capturing the necessary information to repeat and understand data analysis. Then, of course, got stuck. The Galaxy community is actively involved in helping the ecosystem to improve the sharing scientific discovery, improve and sharing scientific discovery. So there is a whole lot of help that's available over Galaxy. In case you get stuck at some position, there is a chatbot that is available to you. You can directly communicate to the people on Galaxy. You could also mail them that you've got stuck here, and what is the problem, they would exactly tell you and handhold you through the analysis. So that is where Galaxy comes in. You're not directly coding anything, but you're still getting the advantage of feeding in your data. And then the analysis is done based on the backend programs that are available at the Galaxy. Right? So this is the advantage. So the paper that we're going to discuss today is this one. This is complete genome sequence of 8 methicillin-resistant Staphylococcus aureus strains isolated from patients in Japan. This is a paper that had come recently. And basically, we take this data because it is simple to explain. I don't want to lead you into a very complicated analysis the first day itself you come to Galaxy, because then some of you would be put off so much that you may not use it at all. So therefore, we take a simple data set of 8 methicillin-resistant Staphylococcus aureus strains, and that is what we're going to analyze. So these are nanopore data. These have been sequenced using nanopore. And then what we're going to go through is to go through the data cleansing process, then of course the quality control, and then of course we move on to assembly. And after assembly, we move on to finding whether there are antimicrobial resistance genes in these bacteria or not. And then of course, we're also going to do a bit of annotation using PROCA. Right? So these are the things that we're going to do today and tomorrow. We'll start today, and then of course we can continue this tomorrow. All of you need to create an account in usegalaxy.org for the analysis. And probably the first thing that we can do is to upload the data at least today, because and when you show the analysis, it is not possible to show it real time because it takes time. So what I'll do is I'll do my analysis and show you the PowerPoint slides of that analysis, and you can follow it up later with your own systems, right? And one of the advantages with Galaxy is that it helps you develop the workflow. So once you're done with your analysis, you can link the steps together into a single workflow. This workflow can be, shared with other people. And now the next person, what he has to simply do is to feed in the initial data. Every step will be then followed as a pipeline. And finally, you'll have the results at the end. So this is something that is, again, advantageous with Galaxy. So once you standardize your workflow, you can use the same workflow every time you want to use it, right? And there is no problem with the data. So here you are. So what are the questions we're answering today? One, how to check the quality of MinION data. Second, how to perform an assembly with MinION data. What species do I have and what is its sequence type? Which resistance genes are there in the genome that we're going to upload? And where are the genes located in my genome, right? So this is what we're going to answer today and tomorrow. Today, of course, we'll start. We still have some 14 minutes left. So we can start today. I will give you a basic introduction and you can upload your data today. And tomorrow we continue with the analysis, right? So here you are. If you go to usegalaxy.org and you can create your account. Even if you have not created your account, you can just have a look as of now. This is the interface that you have at Galaxy here. And if you look at it, there are three panels to your Galaxy interface. This one here is the tool shed where you have all the tools and you can search for the tool that you want to use. And then the right hand side here, you have the history. You can name your history as you begin so that you know what is the analysis you're doing. And then whatever steps you perform here, Neha, your mic is on, right? And whatever steps you perform here will be enumerated here. I'll show you a demo of this from my history, and tomorrow, of course, we will continue with this, right? So here you are. This is your history part. You can use this plus sign and create a new history. Neha, your mic is on while you're not speaking, but it shows some background noise, right? Can you unmute yourself, Neha? Right? And then, of course, if you move along, this whatever steps and whatever analysis you do here will be shown up here, right? So here you are, if you see. So this is your history panel. This is your tool shed. And in the center is where you will be able to see your data and perform the steps or the analysis that you want to do. So once you load a tool here, the inputs requirements for that tool will come up here, and also the conditions that you want to put up, whether you want to filter, whether you want to, change the way you are, whether you want to change the format of your data and so on and so forth, would come up here in the center. And I'll show you how that comes up as we move along. So here you are. This is your workbench. So in the workbench, you can workbench sequentially catalogs the steps of the workflow. And this is your tool shed here on the left-hand side, the tool shed. And this is where you have the list of tools that you can look for and you can use as you move along. Right? So here you are, and you move on further. So first thing is to basically give a name to your history. So you come up here in the history panel and you click on this pencil button. So pencil button is for editing. You click this pencil button and you give a name to your analysis that you want to do. Usually this is a descriptive name so that when you come back, you are able to identify what you have been doing. So for example, here we are looking at nanopore data for MRSA. So I name it as nanopore MRSA, right? Then you move on and you come to this side here. You have the upload data button from where you can upload data. There are various options of uploading data. What we will do is we'll use a URL to upload the data. So the data for us is at Zenodo. We'll use that to upload the data. So here you are. You can upload data from here. And then, of course, you click on this. So the central panel will now open up and it will ask you to upload the data, right? And you have these methods. You can choose local files. If the files are there in your computer, you can browse over and upload the files. You can choose remote files through your connections. You could paste or fetch data. If you have a URL, you can directly paste or fetch data from here. Once you have pasted your data, your URL here, which you're going to do today, we'll say start, and that will allow you to now fetch data from the URL, right? So this is what we're going to do today in the limited time that we have. And then, of course, tomorrow we begin with the analysis. So you'll use this paste and fetch data. You'll use the URL here. So let me also tell you that there is a help available for you. As I said, Galaxy is an interactive. Why is it my tool panel is not coming? Let me minimize this. Something happens to this when you're using WebEx. You don't see. So I was working on R here. So here you are. So here is my tool panel. And here is the help, right? So I'll give you in the chat. So Galaxy is nice in that to handhold new users, it gives you a lot of help, right? So now how do I come back to you all is the question. Oh my God. I think this one here. No. Share, I think no. And no, and I'm sharing my screen, right? So where I am right now, I'm here in this one, right? And how do I come to chat here? I don't get it. Just a minute. Where do I get to chats? Chat. I don't find the chat anywhere. All right. So
Right bottom.
right bottom. No, I don't see it here. Just a minute.
Sir,
Let me
by
just.
minimizing your screen a little bit, you do.
By minimizing the screen, let me do that also and see. Or maybe I can stop sharing and then I'll be able to see you all, right? So just
Yes.
a moment, I'll just stop sharing and then I'll be able to see you all and then I can talk more. You are sharing your screen, stop sharing. Yes. So now I have it in chat and I'm giving you the link for this for everyone. So you can have a look at this and basically I'm giving you this because the data is mentioned here that you need to upload, right? So I hope it has gone in the chat. You will be able to see the link, right? Can you see the link, class?
Yes, sir.
Okay, good. So, I mean, it must be there with everybody now. So let's get started again and let me share my screen again so that you can have a look. So here you are,
Sir,
share.
we haven't got that one.
In the
Oh,
chat it is. It is in the
no, sir,
chat.
we have not got that link. It's not visible there.
Who got the link? I sent it to everyone. Just a minute. Let me stop sharing again. I sent it to everyone. It should be in the chat. Maybe you're not. Okay, maybe. Did you get it now?
Yes, sir, now it's
Yes.
visible.
It
Yes.
wasn't sent. I don't know how come somebody said he got, but it wasn't sent. So now you would get it. Okay, so good. So let's get back and so if you click on this link here, there will be a link for the data, which is with the Zenodo website. You can copy that link and you can come back here, right? So let me now share it again, and then here you are, and you share, and then yeah, right? So let me now come back to Galaxy now. And how do I come back to Galaxy here? Let me minimize this and I come to Galaxy here, right? So if you see, this is my Galaxy, right? You would be able to see my screen, I guess. Everybody, can you see my screen?
Yes, sir, it's visible. Yes.
So this is my history where I've already done the analysis. So if you see here, I've uploaded the data here. The first step, then I've uncompressed the data, then I've filtered the data, then I've used nanoplot to do the analysis part. Then nanoplot, several files are there. Then after that, I've used fly for alignment. And after fly, I've used bandage to get the final assembly. And then of course, I've used fast for assembly analysis for assembly quality. And then proca for the you know the annotation. So we'll do these steps tomorrow very quickly, right? And probably I'll go through the presentation again because to do it online is not possible because each of these steps takes time. But for you, what I'll do is I'll create a new history and at least I'll show you how to upload the data before we close today, right? So here you are, you create a new history, you click on the plus button, you will get this unnamed history. So let me just put it as next gen, right? And nanopore. Right? The name has to be unique. And you say save, right? So here there is nothing as of now, if you see, right? Now I come to upload data here. So everybody, you please follow. I'm doing it because I want you to upload the data now, right? So open the tab for the tutorial that I've given you and you go to upload data now. So you click here. So here you are. This is where you can add your data. And what we're going to do is we're going to use a URL to add data. So we'll say paste and fetch, right? So you say paste and fetch. Here you are. This is where you can add your URL for the data. So we'll go back here and we'll fetch the URL for the data. So this is the URL. This is on the zenodo.org, right? You can copy this here and you can paste it here, right? So you paste this here. Everybody with me, class?
Yes, sir. Yes.
Good. So you're doing it, right?
Yes, sir.
Paste it
Excuse me.
here and then you say start. So you click on start here, right? Right? And then you can say close. And if you see on the left-hand panel in the history panel, now you see there is a buffering and you can see the name of the data is showing up here. So this is what is happening is that now the process of loading the data has begun. When the process is over, this will turn green. And that is when you can start with the next step of the process. You follow class, everyone?
Yes, sir. Yes, sir.
Okay.
Actually, we are doing it side by side.
Yeah, that is how it is to be done. But of course, tomorrow we'll not be able to do side by side because this will take time. Some of the analysis will take real long time. So I was doing it again for you all today. So I uploaded my files for nanoplot and it took almost 5-6 hours for nanoplot to work because it was queued up and it just didn't go into the actual process for a very long time. So it depends on how busy the server is and how big your data is. You will have a lot of time for the analysis. It will take a lot of time at each step. And I've already told you that the assembly process is going to be the most time and memory intensive, right? So what I suggest you is that in case you've not created an account here, create an account on galaxy.org so that your history remains saved. If you're logging through a guest account, once you close the account, once you close your Galaxy, it will not show up again. So after the workshop, we close at 8. Then you can start your, creating an account and doing the first step again. I've already given you the link. Copy that link somewhere else because I think the link will close. Once I close the WebEx, it will not be seen.
Excuse me, sir.
Yeah, the data is link is shared. Yes, tell me.
Sir, actually, no, sir, in Galaxy account, I am not able to register. Actually, it is not opening .org.
You could choose some other galaxy.eu. It is usegalaxy.org, right? If you
So
see
that
here,
used, but actually, it is not opening in my system.
so maybe you could use, there is other instance of Galaxy, galaxy.eu. You can take that up maybe.
Okay,
Arpita,
and Galaxy, sir, other tool is also there
which
because
other
I'm
tool?
other for accessing actually for my data because it is not online one.
Which other tool you're talking about? We are talking of Galaxy. This workshop is on Galaxy. You could use Taverna also, but
I tried many times, but it is not opening.
I mean, this is an individual problem. You could check your network. Maybe you could have blocked some pop-up or something in your settings of internet. Everybody else, Galaxy is opening? Class? We have
Yes,
20
sir.
people
It's
here.
opening. Yes.
Yes, sir.
Yes. So the server is not down. It is not a problem of the server. It could be your individual problem. So you need to check your settings and then work it out. Okay.
Yes, sir.
So let me take you through very quickly through what we are going to do tomorrow. Before we close, we'll
Excuse
have
me,
a
sir.
yes.
Uh, what kind of a data we can upload? Any kind of a data means a
No,
Word note like that.
no. So this is basically like this one is .fastq file if you see, and this is a compressed file, right? So Word document you can definitely not upload. When you're doing programming, Word file is not something that we work with. Okay, it will always be a .txt file, the notepad file, right? Most of
Okay.
these would be okay.
What kind
Word
of
file is not something that is
sequence?
readable by programs.
What kind of a sequence? mRNA, RNA, DNA, any protein, anything we can give.
Yeah, so that depends on the analysis that you want to do. If you're doing an RNA analysis, the pipeline will be very different because the tools that you're going to use are going to be different. At the base of it, what you're doing is you're uploading a FASTQ file. I told you what is the format of a FASTQ file, right? There are 4 lines. You have the @ sign at the beginning and a unique identifier. The second line is your sequence. The third line is again a plus sign and the identifier may be repeated or it may be absolutely empty. The fourth line is the quality line, right? So you would know what analysis you're going to do. Based on that, what files you need to upload is something that you would know by yourself. If you're doing a protein sequence analysis or protein structure analysis, then it would be a PDB file, a standard PDB file. So I've already told you the formats have to be standard. You cannot just
Okay.
upload anything or everything. Okay.
Okay. Okay, sir. Thank you.
So I guess everybody clear?
Yes, sir, it is clear.
Okay, so maybe what we can do is we can stop here, and tomorrow we continue with this. I'll show you mostly through PowerPoint what all we have done, because doing the analysis itself would take a lot of time. And if you are interested, you can follow the instructions here in the link that I've given you. They have exactly defined what is the next step that you need to do to get your results, right? So you could do that. But of course, tomorrow I'll show you stepwise. I have taken the screenshots from the previous analysis, and I'll show you the screenshots of how this can be done, and also give you some explanation of the data that is generated from here, right? So we close now, and we continue with this tomorrow. Okay, class?
Okay, thank you so much.
Questions?
Okay, sir.
Thank you so much, sir.
Yeah, you're welcome. Everybody is welcome. Joy.